students, welcome today. We're going to have two speakers. We're going to start off with Jacob Steenrick, who is a graduate student at the University of Vanderbilt in Antonin Roukis's lab. I was just chatting with Jacob and learning about his dedication to science, but also his interest in science communication, which you can see more about on his website. I think maybe you might have a few of those in your talk, Jacob. Today he's going to be telling us about his work on Aspergillus hybrids, which was recently published in Current Biology. And with that, I'll just let you take it away.
Oh, awesome. Oh, I think the host has disabled participant screen sharing. Okay, awesome. Okay, can everyone see my screen?
Yes, we can see your screen.
All right, fantastic. Okay, thank you everyone for being in attendance, and thank you to Professor Blue and Brown for hosting this and allowing me to give this talk. It's a true honor. So I recognize that most probably are not familiar with who I am, so I wanted to take some time and introduce myself. I think who I am is largely shaped by the amazing people that have helped raise me. So this is an image of my family at my sister Emily's wedding some time ago, and she's now married to Josh. And one thing that's really awesome about this very supportive and amazingly talented family is that it's growing. So the newest member is now Liberty Rose, my niece, my goddaughter. So if you hear a baby crying, please note that I am living in a household with a newborn. One of the things that my family has supported me doing is also some of my side projects and the other interests that I have, such as art communication, as Professor Ballou had mentioned. One project I'm involved in and have started from the ground up is this Art for Earth initiative, or the SciArt initiative. And what I'm really trying to do is allow people to purchase with purpose. So 100% of the profits that the shop makes go toward global conservation efforts. So it's really secondary that we're selling stickers, poster prints, mugs, whatever. It's really about using art as a mechanism to raise awareness and, in a very fantasized sense, immortalize these endangered species. So these are some of the endangered species that I've highlighted out of a total, I think, 20 or so portraits. I am, of course, a mycologist, and so I do also make portraits for the species that I think are interesting or that I like in some manner. So these are also some fungal themed portraits. And very recently, I had the great honor of writing a perspective piece about the arts, sciences, and how their whole is greater than the sum of its parts for yeast, and do the cover of that journal as well. So since opening in August, the shop has earned just over $1,000 for wildlife conservation, and I really look forward to continuing to promote this Art for Earth initiative to do what I can to help conserve wildlife. Okay, but today, of course, it's really about the science that I've done, and I've been lucky to be working with Antonis Roukis. He's my PhD advisor at Vanderbilt University, and Antonis's lab has like three main pillars of research. One of which is studying evolutionary relationships and the parameters that influence these inferences. Another is to study the evolution of human pregnancy. And the third pillar is the evolution of yeasts and molds. And this is really where most of the work that I've done falls, in this yeast and mold evolutionary subject. More specifically, I'm very interested in the rapid tempo of evolution among fungi. So some studies that I've done have investigated ancient rapid evolutionary events in fungi, more recent rapid evolutionary events, or developing methods and software to help enable the study of these processes. So when it comes to ancient rapid evolutionary events, one project that I've done is investigating this budding yeast lineage or genus called Hensiniospora. And there's two main lineages, one that evolves very fast, one that evolves a little bit slower. And it's commensurate with how many DNA repair genes and cell cycle genes they've lost. These genes that are very important to genome and cellular integrity that were once thought to be evolutionary resistant to change, but this and other papers by other groups are sort of overturning that notion. As for the software I've developed, I've had the experience of developing diverse types of software. Some of them do very simple things such as OrthoFisher, which helps extract genes in an automated fashion using HMMs. And then ggpubfigs, which helps create publication quality figures, and importantly, using colorblind friendly color palettes so that it's accessible to the broadest audience. Some of the other software I've developed is a bit more complex in what it does, such as ClipKit, alignment trimming algorithm, and the sister toolkit, PhyKit, which is a very broad set of functions for processing and analyzing multiple sequence alignments and trees. I want to say a little bit about ClipKit just to give you a glimpse into some of the software I'm developing. And ClipKit falls into the broader phylogenetic pipeline where you first align your homologous sequences, trim that alignment, and then reconstruct their evolutionary history. So ClipKit is part of that trimming the alignment step. The whole reason that people trim alignments is because highly divergent sites are thought to be caused by erroneously inferred site homology and saturation of multiple substitutions. So those highly divergent sites are often trimmed away. And this has been common practice for nearly 30 years, ever since Lake had written about it in 1991. More recent analysis coming out of Tan et al. In 2015 had demonstrated that most multiple sequence alignment trimmers actually resulted in worse single gene phylogenetic inference. And the take-home message was that the more aggressive the trimmer, the worse it performed, which they didn't explicitly say this, but this suggests that most current methods are actually removing sites with phylogenetic signal. So I really felt like there was this need for an alternative alignment trimming strategy. And rather than removing uncertain sites, I decided, well, what if we just focused on sites with phylogenetic certainty or with phylogenetic signal? And that's really the crux of what ClipKit does. There's various modes that ClipKit uses that have very nuanced differences between them. So I won't explain the various modes, but I will show you how well it does perform. So the performance is ranked where first is, like first place, and then last place is last place. So the lower the number, the better for the y-axis. And then they're arranged from left to right based off of best performance and worst performance. So this is in simulated data sets. So no trimming is always going to perform the best in a simulated instance. And then these sorts of trends where ClipKit with the blue stars above it performing so well were also observed in empirical data sets. And this is a ranking system based off of tree topology accuracy, tree support, and accuracy of branch length estimation when using those ClipKit trimmed alignments. Okay, so today I'll be focusing more so on this more recent rapid evolutionary event that has to do with hybridization among fungal pathogens. I first want to talk about a very classic hybrid, a hybrid that everyone is familiar with, and that's the mule. The mule is so iconic because it's been used to bridge physically distant cultures and acquire physically distant resources. And this is in part due to a phenomenon called hybrid vigor, wherein the mule has some of the best traits from both of its parents. So from the donkey, it has strength, patience, and endurance, and from the horse, athletic ability and speed. Biologically, the reason that a mule is even possible is that because the donkey and the horse, despite looking a bit different, they're actually pretty genetically similar to one another. They're less than 1% diverged. Yet that divergence is enough to make the mule, the product of this hybridization event, sterile. So it's an evolutionary dead end, so to speak. Now there's different types of hybridization events. Some are hybrid speciation events, and this is observed in fungal plant pathogens like Zymoseptoria species. Ava Struckenbrock has a lot of work on that. And then yeast human pathogens, such as the work from Tony Gabaldon investigating various Candida species. And then there's introgressive hybridization where smaller loci can be exchanged between different lineages. So today we'll be talking about a hybrid speciation event wherein two different species of fungi, they came together and they formed a new species. And that's where the main part of the talk begins, when two become one, the hybrid origin of a filamentous fungal pathogen. So for this part of the talk, I'll be talking first about Aspergillus fungi, introducing the players, And then we'll talk about the genomics of the hybrids and the phenotyping of the hybrids. So Aspergillus, they're pretty deadly fungal pathogens. They pose a clinical, economic, and social threat to human welfare, and they're responsible for over 200,000 infections a year. However, not all Aspergillus species are known to cause disease in animals or in humans. So this suggests that pathogens have evolved multiple times when you look across the phylogeny of Aspergillus species, where the animal pathogens are denoted with the maroon circle. So one of the big questions that Antonis Rokas and many others in the lab are interested in investigating is, what are the evolutionary forces that lead to some species being able to cause disease, whereas others are harmless or even beneficial to human welfare? So to investigate this, we decided to take an in-depth look at just one species, and that's Aspergillus nigilans. Aspergillus nigilans primarily infects those with chronic granulomatous disease, commonly just referred to as CGD, but it's recently been associated with some other lung-related diseases. So our approach to study Aspergillus nigilans as pathogen was to acquire Aspergillus nigilans from the hospitals, grow them up on plates, do various phenotyping, and sequence their genomes. And we were told by the clinicians, working in the hospital, that these were Aspergillus nigilans, and this had been deduced using microscopy methods, which is commonly employed in various parts of the world as a way to identify what a patient is infected with. However, upon genome sequencing, we had found that their genomes were not like the typical Aspergillus species that we usually see, because a typical Aspergillus species has around 10,000 genes, a genome size of around 32 megabases, and very few duplicated genes, which is shown on the z-axis, these BUSCO genes, these genes that are otherwise thought to be near universally single copy. But these clinical isolates shown in purple, they had twice the genome size, they had twice the number of genes, and many, many of these genes were duplicated. So this led us to believe this isn't actually Aspergillus nigilans. We should conduct some molecular phylogenetic analysis to discern what these clinical isolates are. And molecular phylogenetics pointed to each hybrid, or each isolate, clinical isolate rather, having two copies of taxonomically informative loci, beta-tubulin and calmodulin. And each copy belonged to a distinct clade of Aspergillus species. So just to show you where one strain is and how it falls into two different places in the phylogeny, this arrow is pointing to strain ASFU 2033. And you can see it has this one blue copy and this one red copy. Now the blue copy is Aspergillus spinulosporus, as it's monophyletic with that species. And then the other copy, we're not really sure what the other parent is, but it's a close relative of Aspergillus quadrilineatus. So in summary, this molecular phylogenetic analysis pointed us to these clinical isolates being hybrids, and also pointed us to what the parental species is, or a close relative of the parental species. With additional genome sequencing, additional genomic analyses, we were able to determine that the new parent that came from this hybridization event is Aspergillus latus. And again, it came from the hybridization of two different species. So we've come up with a model for how we believe this might have happened, where Spinulosporus and this Aspergillus quadrilineatus, like species of Aspergillus, likely underwent cellular fusion or plasmagamy, forming a heterokaryon that then underwent nuclear fusion or karyogamy, resulting in Aspergillus latus, a diploid allopolyploid. Now, interestingly, unlike the mule, which is an evolutionary dead end, Aspergillus latus can produce both asexual spores and sexual spores, both conidia and ascospores. So now that we had the genomes of these hybrid species and we knew that they were hybrids, we next wanted to sort of tease apart which part of the genome came from which parent. And the way that we did this was to leverage sequence divergence of each gene to a known parent, and then plot their frequency along sequence divergence as the x-axis. So what ends up happening with this type of analysis is one set of the genes will be very similar to the one parent that you're comparing it to. And then there'll be a second set of genes that are very different from the one parent that you're comparing it to. And these two modes, this bimodal distribution reflects the genes coming from each parent. So you can split them up in a pie chart and assess how much each parent is contributing to the extant genome. And if we were to think about this as a mule, it's almost as if we're able to identify the genes that came from the donkey versus the genes that came from the horse. So for the hybrids, what we found is pretty much both parents are equally represented. We saw that bimodal distribution that one would expect for a hybrid, and we were able to tease apart which part of the genome came from which parent. Just to highlight one strain, I'll highlight the same strain that I highlighted earlier during the molecular phylogenetics. This is ASFU 2033. You can see that pie chart. Both of those genes are pretty much present in equal proportion. So now that we had roadmaps for the genomes of these hybrid species, we did analyses of gene pairs. We looked for recombination. We looked at copy number variants, or CNVs, duplicated and deleted loci. We looked for evidence of aneuploidies, and much to our surprise, they had a high level of genetic stability. We didn't see a lot of degradation throughout the genome, which is typically observed in hybrids where the hybrid is an unstable state of being. So with this, we were able to also do other analysis since we had the genes that were coming from each parent. And we also asked, how different are these two parents? How different is Aspergillus spinulospores to this Aspergillus quadrilineatus-like species? And they're roughly as different as a human is to a lemur, or roughly 8% diverged. And so for the animal kingdom, that would be wild. For the fungal kingdom, that's roughly on par with what we would expect for fungal hybrids. Next, I want to go into the phenotyping of the hybrids. And I think the take-home message here is that the hybrids exhibited wide phenotypic variation. So we examined virulence, interactions with human immune cells, drug susceptibility, growth in various conditions, all sort of biomedically relevant phenotypes. And when we summarize all of that information, we noted using principal component analysis, or PCA analysis, that there was a lot of strain heterogeneity within Aspergillus latus, as denoted in purple. We also found that they were very similar to one parent, Aspergillus spinulospores, as opposed to the other parent, Aspergillus quadrilineatus. Now, this may be because Aspergillus quadrilineatus isn't exactly the other parent. And so, with additional genome sequencing of close relatives of Aspergillus latus, quadrilineatus, and spinulospores, we might be able to narrow down what's happening there. One thing that I do want to point out is that they're very different from Aspergillus nigilans, which is shown in the bottom left here where my mouse is. So that's important because in the clinic, they were identified as Aspergillus nigilans, but they're not, and phenotypically, they're very dissimilar from Aspergillus nigilans. So I've noted how there's wide phenotypic variation. Just to give you a more concrete example, these are the Galeria kill curves, so an invertebrate moth model of disease. And we noted that sometimes Aspergillus was the least virulent, such as strain Mo 46149, or the most virulent, such as strain ASFU 1710. So wide variation in how virulent these were in this disease model. In other instances, they were more like one parent than they were compared to the other. So when it came to drug susceptibility, they were more like Aspergillus spinulosporus. And when it came to resistance to hyphal killing by human immune cells, they were more like Aspergillus quadrilineatus. And in both instances, this was the more robust phenotype. So they were less drug susceptible, or they were more resistant to hyphal killing. And this is eerily similar to the situation that's going on with the mule, where it has some of the best traits from both parents. So we hypothesized that this could potentially be some hybrid vigor, and this is something we look forward to continuing to investigate. So I think the graphical abstract nicely summarizes the manuscript, where we sequenced these clinical isolates of Aspergillus fungi. To our surprise, we found evidence of allodiploid hybridization and genome stability amongst these hybrids, amongst these clinical isolates, and that the hybrids, they phenotypically speaking, differed from each other as well as their parents, or sometimes were more like one parent over the other. In a more broader sense, I think the study's importance is twofold. Medically, we show it's important to have accurate isolate identification in the clinic, and also acknowledge that one strain is not really representative of an entire species. So strain heterogeneity is an important component. Evolutionarily, we show that hybridization seems to be a general mode of fungal pathogen evolution. So earlier, I had mentioned how human yeast pathogens can hybridize and how plant pathogens can hybridize. But to my knowledge, this is the first instance of a filamentous fungal pathogen of humans that can hybridize. So with that, I would like to thank the project team, this really fun, collaborative international group. I especially want to thank the co-first author, Abigail Lind, who's now a postdoctoral fellow at the Gladstone Institute over in San Francisco. I want to thank the Rokas Lab, and I would also like to thank the Howard Hughes Medical Institute for their predoctoral funding through the James H. Gilliam Fellowship. And so yes, with that, thank you very much again. I would be happy to take any questions.
Thanks, Jacob.
Thank you so much.
Oh, sorry. Thanks, Jacob. That was fascinating. If everyone would enter their questions into the chat, we'll have questions for both speakers at the end. And so, Bertrand, would you start our screen sharing, and I will introduce the second speaker.
Yeah, thank you.
So our second speaker today is Bertrand Nyuykonge, who is coming to us from Erasmus University in a medical center in Rotterdam. And he is going to tell us about mycetoma causing fungi. Bertrand?
Yeah, thank you, Professor Brown. Good afternoon, good morning, good evening, wherever you are, everyone. Yeah, I would like to thank the organizers for giving me the opportunity to share some work from my PhD project. So here we discuss a short tandem repeat assay for studying genetic variation in Madurella mycetomatis, the main causative agent of mycetoma. First a brief background. Mycetoma, probably a lot of people haven't heard about it, but it's a neglected tropical flesh-eating subcutaneous chronic infection, which is characterized by inflammation, painless tumor-like lesions, and multiple sinuses, which discharge this tiny material called grains, which differ in color depending on the etiologic agent. Most often, it affects the feet and hands, as you can see, like below in the depiction, where you have a sample picture of a normal foot in A and a foot affected by mycetoma. In real life, it would look like this. I hope you can all see my mouse. So you have these large tumorous lesions, and the tiny spots below are the black grains, which are coming out from the multiple sinuses. So mycetoma can be caused by either bacteria, which is then referred to as actinomycetoma, or it could be caused by fungus, which is then referred to as eumycetoma. But in our group, we mostly focused on the fungal type of mycetoma. So there are quite a dozen, like so many causative agents of eumycetoma. I think currently we have about 20 or more. But the main causative agent of eumycetoma is the fungus Madurella mycetomatis, which is which would be the focus of this discussion, and also is the main organism that we study in our group. So the issue with mycetoma being a neglected tropical infection is like the global burden is entirely not known, because it is mostly sort of prevalent in poor rural communities, so people do not have access to healthcare facilities, so it's difficult to actually know the numbers. But to help with quantifying the global burden, it is necessary that the epidemiology and that much work is done with regards to epidemiology as well as mapping. And to do this, we need typing tools. So, but why do we actually need to type? This is, in essence, to give us clues with regards to its pathogenicity, the transmission routes, because at the moment, the method of transmission is not entirely clear. It is hypothesized that people get exposed to the fungus by exposure to a thorn, which might have the fungus, and then the fungus gets access to the body. And if with typing, if we're able to probably type environmental and clinical isolates, we might be able to locate, probably the route of transmission or where it is located, and this would help us with designing prevention strategies which are effective. So there have been quite some studies with regards to typing, and these studies have used pattern-based DNA techniques such as random amplified polymorphic DNA analysis, restriction fragment length polymorphisms, and also amplified fragment length polymorphism. And the latest one, which was done in 2018 in our group, was done using VNTR. And these techniques had a variety of issues. First was that the reproducibility was very low and the discriminatory power was not that good. Hence, there was a need to develop an assay which is highly discriminatory and which is reproducible to enable the transfer of information or data between various research groups or various labs. So short tandem repeats assays offer us this opportunity. First, short tandem repeats, or also called microsatellite or simple sequence repeats, are repetitive DNA motifs which range from 1 to 10 base pair. And they, for instance, they could be di, tri, or tetranucleotides. They're quite variable through and very abundant in the genome of most eukaryotic organisms. And they are often predominantly within non-coding regions, though there are a few cases in which tandem repeats have been reported in coding regions. So basically, STR assays or short tandem repeat assays are determined to measure the exact number of repeating units. For instance, if you look on your right, or on my right, if you look with the trinucleotides, the GCA repeat unit, we see that there are 5 repeat units in this short sequence. So how STR assays work is that because these repeat units are always sort of highly variable between strains within a given species, this variability is used to discriminate between strains. So, like I mentioned, but why are STR assays so popular? They're popular because STR markers themselves are quite abundant, and they're evenly distributed in the genomes. You don't need lots of DNA. You can use as low as 0.5 nanogram per microliter. You can easily detect them by PCR. You can also multiplex them, which improves the throughput. They're highly reproducible, which solve the problems associated with pattern-based techniques. And they're also highly discriminatory and specific, and also it's cheaper and easier to analyze the data. So based on these advantages, our aim was to develop a robust and highly discriminatory short tandem repeat assay for studying genetic variation in Madurella mycetomatis, which we abbreviated MmySTR. So to do this, we had this experimental setup. So first, we selected STR markers from the reference genome of Madurella mycetomatis using tandem repeat finder software. And then we design primers. First, I have to mention that we consider a number of criteria in the selection of STR markers. One of such criteria was to make sure that the flanking sequences, were highly conserved, so as to ensure that we could design some primers. And we also selected loci with the highest number of repeats. So with this, we designed some specific primers. Then we did some DNA isolation, and then we validated these STR markers. First, we did that in singleplex and multiplex. However, to enable high throughput, and high resolution, we needed and also to enable high resolution, we needed to use capillary electrophoresis because the resolution is better than that of gel electrophoresis. So to do that, what we did was we labeled the markers that we had selected. So how we do that was we attach a fluorescent probe, which has a particular color on each forward primer, and following amplification, then the PCR product became fluorescent, and then yeah, this could be detected using capillary electrophoresis, as I mentioned. I would explain how this works in subsequent slides. So based on this criteria, we had a panel of 11 markers. We had 3 dinucleotide markers and 4 tri and tetranucleotide markers, and the repeat units are shown here, while the label primers are shown here. So the FAMV, NATE, and TET are the fluorescent probes, while the allelic range is here. So we see that most of our markers were sort of situated in intergenic regions. So with this panel of 11 markers, we needed to see if they do work. So here I would explain with a brief example. So we selected 3 isolates of Madurella mycetomatis. That is MM55 from Sudan, PU1 from Mali, and PA from Algeria. And following amplification, when we ran that on capillary electrophoresis, we could see like peaks. For instance, these peaks here represent the size of each marker. So here is the size of the band. For instance, with the red, if you can see my mouse, the red marker here is for MM55 is 141 base pair. So with these peaks, if the peaks differ between isolates, it would mean that the marker is able to distinguish between the isolates. For instance, the red marker here is not able to distinguish between MM55 and P1. However, it's able to distinguish between PA. But then if you look overall, we see that the black marker, which is you can see here for MM55 and P1 as well as PA, is able to distinguish between the 3 isolates. So you can also see in this figure that, for instance, with the red marker, you have a large peak here and a tiny peak here. The tiny peak is as a result of polymerase dependence activity, which, for instance, if a sequence ends with a G, a polymerase, the tag would normally add a poly A tail. That's why we have an extra peak here, which is 1 base pair different from the main peak. So yeah, so we did same thing, similar thing for the tetranucleotide markers and the dinucleotide markers. So with this 11 marker panel, so from the peaks that I showed in the last slide, based on the fragment size, we could extrapolate the number of tandem repeats from the fragment size. And so for each marker, we had this 11-digit code, which was particular for each isolate. So we see that, yeah, like for the trinucleotide marker, that's MmySTR3A, which was all in blue, it wasn't able to distinguish between any of these isolates. So we see that the number of tandem repeats are exactly the same. But if we choose another one like MmySTR2A, for instance, that's a dinucleotide marker, we see that it's able to distinguish between the 3 isolates. So what we did is we validated this 11 marker panel in a large collection. So here in this figure is a minimum spanning tree of this, which we obtained using this 11 STL marker panel. So what we did was we used 120 isolates and we ran it on our 11 marker panel. And we saw that these Madurella mycetomatis was actually very genetically divergent than previously thought. So in this figure, each circle, for instance, represents a genotype, and the size of the circle is directly proportional to the number of isolates in that genotype, while the colors represent a country. So with from these 120 isolates and using this 11 marker panel, we were able to obtain 108 genotypes, which led us to a d-value of 0.997. The d-value is a Simpson diversity index, which is basically a measure of diversity, or the probability that typing assay would assign different genotypes to two randomly chosen isolates in a given population. So previous studies using LFP had shown that certain genotypes tend to be associated with lesions size. So we investigated with our assay to see if we would find this association in which any of the MmySTR genotype would correlate with lesion size. Unfortunately, or as expected, because most of the markers were not within coding regions, we saw no correlation of genotypes with lesion size. So here, each circle still represents a genotype, and the colors represent the size of the lesion. So the green is the small, the yellow is the medium, and the red is the large. So based on this, we concluded that our MmySTR genotypes had no correlation with lesion size. Furthermore, we compared our assay with previously designed assays. First, with the LFP, when we projected our data on the LFP, we saw that our assay was more discriminatory. So it's depicted in these figures. So with LFP using 33 isolates, we could only obtain 3 genotypes, which are highlighted by the colors. So we have the green, red, and blue, which corresponds to LFP genotypes. However, when we take these 33 genotypes and we run on our assay, we obtain 30 genotypes, showing that our assay was more discriminatory than the LFP. So another assay which I mentioned, which was recently developed, was the VNTR, which sort of more or less discriminate, isolate based on variable number tandem repeat. So previously using 77 isolates, 12 VNTR genotypes were obtained. However, when we use the same isolates and run it on our assay, we obtain 73 genotypes, showing, also confirming that our assay was more discriminatory than the VNTR. So based on this, we conclude that our MmySTR assay was superior to the LFP and the VNTR. So with this, So with this, to confirm this, we look at the Simpson diversity index for all of the methods mentioned, and we saw that with LFP, the Simpson diversity index was more or less 0.5, while with VNTI was more or less 0.8, and with the MmySTR was almost 1, showing that indeed, confirming that the MmySTR was indeed superior to the other pattern-based techniques mentioned. So with this, we had the following conclusions. So based on this, we concluded that Madurella mycetomatis is quite genetically divergent, and that this genetic diversity may explain the different manifestation of mycetoma or eumycetoma, and also that this genetic diversity is likely as a result of sexual recombination. And we also see, we can also conclude that the MmySTR assay is quite robust and highly discriminatory. And finally, we demonstrated that the MmySTR assay was superior to pattern-based techniques such as the LFP and the VNTR. So for future perspective, what we intend to do is establish the MmySTR assay as a reference method for studying genetic diversity of Madurella mycetomatis isolates. And with regards to potential applications, we hope to determine the global diversity of Madurella mycetomatis using this assay. And because like the first picture which I showed with with the food with quite a number of lesions, we also want to check if it's possible that there could be a mixed infection within a single infection or within a lesion. And then also with this, we could also determine if it's possible to differentiate between recurrence or reinfection. And we also hope to establish the source of infection by typing clinical and environmental isolates. So with this, I would like to thank the following people that contributed to the work. The technicians in our group, Kimberly, Wilhelmine, and also some people from the French Institut Pasteur, the Center of Drugs for Neglected Disease Initiative, and the Mycetoma Research Center in Sudan. And if you're interested to know more about our assay, you could check our publication in JCMI. And also, if you have any material or any Madurella strains or archive or DNA material that you would like to type, you could get to me by my email, or you could get to my supervisor, Wendy van der Sander. And with this, I would like to thank you again for the opportunity to share some of some work from my PhD. Thank you.
Thanks, Bertrand. That's a wonderful talk, and it sounds like a fantastic new assay. So we're going to take questions now. So if you would please enter your questions into the chat. We'll start with Jacob to give everyone time to put questions in for Bertrand, and we'll go back and forth between the two. So feel free to enter questions as soon as you can come up with them. Liz?
Yeah. So question for Jacob. So this is just starting off from the top. Jessica Brown asks, she's struck by the evolutionary distance between the hybrid parents that are still capable of producing viable progeny. And you mentioned that this was something that wasn't really unexpected for fungi, but is very unusual in other eukaryotes. So she asks, why do you think this fungi might be capable of this? Is it because of physiological conservation of mating structures, or is there something else that's driving this?
Yeah, we don't know definitively. Some clues that we have is that different Aspergillus species are more likely to undergo sexual reproduction. So like Fumigatus will not, but Nidulans will. And these are close relatives of Nidulans. So we think that, they might just be more used to this sort of lifestyle. But yeah, it's TBD.
Yeah, I just following up on that. I was really interested in this observation that you had, that there were these duplicated copies of these normally single copy genes. Is there a reason that you could discern that there were normally single copy? That we talk about aneuploiding having fitness costs, but is this hybridization having a fitness cost as well? That might explain that, or? Yeah,
there was nothing that we could tell that was obvious. Like, it's not like they grew differently. The phenotypes varied quite widely, and different measurements or different phenotypes we were measuring came up with different results. But it didn't really seem like fitness was that impacted in these hybrids, surprisingly. Again, this might have to do with lifestyle, where these fungi are used to having multiple nuclei in one cell due to their heterokaryotic lifestyle. So we think that this might be playing into them being more used to this type of genome, or this number of nuclei in a given cell, or this much genomic content.
Yeah, I think that was such an interesting. Did you guys look at nuclear dynamics in your hybrids compared to the parents? Were you able to do that?
We haven't done it yet, but this is on the to-do list. Yeah.
Yeah, great. Jessica, do you want to ask a question for Bertrand?
Great, thank you. So Bertrand, it's really So this question comes from Liz, but I think we can all agree that with her initial statement, that it's really surprising how much diversity you saw in these isolates. And so she asks whether these isolates are primarily from, is it correct that these isolates are primarily from Sudan? And would you expect a similar level of diversity from other locations? Or do you think that something specific is going on in that fungal population there? And also, it looked like, at least in your acknowledgement slides, there was an institute looking for mycetoma research in Sudan. And so do you think that there's some sampling bias or something else?
Yeah, thanks for the nice question. I think, it's right that majority of the isolates we had were from Sudan. Yeah, that's because we've been collaborating with the Sudanese for the past 25 years or so. So we tend to have more isolates from there than any other place. Yeah, but after we developed this assay, we've been trying to contact people, to actually get more isolates from other areas. But not shown here, but I think that we would still see the same genetic diversity from isolates from other areas. I might not have mentioned it, but there's some work we're doing. It's still quite preliminary, but we see the presence of like the possibility of sexual recombination in this organism, which might suggest that it's probably sort of reproducing in the wild, but now we don't know the natural niche at the moment, so it's quite difficult, but definitely interesting to look forward to the future with regards to what we would see.
Yeah, that's fascinating. Thank you. And Anna Solmecki asks a similar question, which is, do you see different genotypes within a single patient? And then what happens when you culture these fungi in the lab? Do you frequently see changes in their tandem repeats or other genotypic changes?
Thanks for the question. With regards to I think, sorry, I lost track of the question again.
The first part was, do you see different genotypes in a single patient?
Oh, yeah, that's right. So far, we haven't seen that. It's also some data which we just generated last week, in which we selected multiple, because the causative agents normally doesn't appear like a fungus. It's always like the grains, the black grains. So what we do is we always, to type directly from the patient, we always extract these grains, and then we isolate DNA from it. So we got some data last week in which we had 11 patients and we sampled them from different parts. But what we saw was that for each patient, irrespective of the sampling part of the body, they had the same genotype. So far, we don't think that we haven't seen an example of a mixed infection at the moment. Then, with regards to culturing in the lab, because this organism does not form spores. It's possibly possible in nature, but at the moment we've not been able to generate any spores in the lab. So we check for the stability of these markers to see if the genotypes would change with when we culture them in the lab. So what we did was we selected some few isolates and we sorted them about 155 times. But we didn't see any change. We even tried to see if they would mate, but it doesn't work. It's quite difficult to check for stability or any evolution if it's non-sporulating, or if you do not. If we would know like the natural niche, then we would be able to try to mimic the conditions in the lab. But we tried several times and with no success. So but we would keep on trying to see if we can generate some spores.
So a naive question for me, and that is, do you know whether this organism is naturally haploid or diploid, or is it not known?
What we see is it's not entirely clear, but based on preliminary results, I would say is most of the time it tends to, yeah, I would say based on the sexual cycle, it seems to have the same sexual cycle as Aspergillus, based on DNA analysis. So we see a similar sort of sexual recombination pattern, but still, if you cannot make them generate spores or see some sort of sexual cycle, it becomes difficult to really, to really draw a conclusion.
Great, thank you very much. Liz, do you want to take a few more questions for Jacob?
Yeah, I think we've got some interesting conversations and parallels here with questions in the audience for Jacob. Shannon and Matthew are both asking sort of what's happening in the clinic versus what's happening in the environment. So Jacob, just building off of what Bertrand just said, do you think that these are hybrids that arose in the environment, or are they specifically because they're in the context of a patient? And is there a particular patient population that might be, particularly susceptible? Matthew's highlighted the CGD population.
Right. Yeah. Thank you for the question. We anticipate the CGD population is most susceptible, and so to further understand whether or not this is happening in the patient or outside of it, with the current sampling that we had for the manuscript, which was 6 samples, we couldn't address the question. However, more recently, we've conducted population level sequencing. We now have 40 different isolates, but we're unsure how many of them are even hybrids. So they've all just been typed as nidulins in the clinic. And so we hope that with that sequencing in hand, we'll be better poised to address the question of where did hybridization occur, but also how many times did it occur? Is this a single origin event, or have there been multiple hybridization events?
Yeah, absolutely. And just building on that, so the second question from Alex, Sora is asking, do you have plans looking at hybridization in other filamentous fungi in addition to nidulins?
Yeah, we definitely do. I would say that we kind of discovered these hybrids serendipitously, right? We were trying to do a study on nidulins, but it just wasn't nidulins. So I would say that we are definitely open to it and taking lessons from those that have, set the foundation for us of like what good experiments should we even conduct. But I don't think we're explicitly focused on hybrids in the clinic.
Yeah, more interested in understanding nidulins specifically.
Mm-hmm.
The last question I have for you is one from Bertrand, actually. What he's wondering about is, because you mentioned in the phenotyping that there was increased resistance to antifungals. Was this all classes of antifungals, or was it a particular class? And do you think that was, I'll just add on to that, do you think that was related to the duplicated genes that you observed, or was there some other mechanism that might be going on there?
Yeah, that's a great question. We sampled a diverse set of antifungals, and this was really the work carried out by our collaborator, a fantastic collaborator, Gustavo Goldman. And so he did all that work. It spanned multiple classes. I'm not sure if we got all classes of antifungals, but we got a lot of the classics that would be used in the clinic, such as caspofungin. Mm-hmm. When it came to the phenotypes that we observed, we noticed that they were more often like one parent over the other. And so we think that there might be some genotypic dominance thing going on here, rather than like an additive genetic effect where they're, as robust as both Quadrilineatus and Spinulosporus. Yeah,
that's really interesting. Have you got plans to do any transcriptional profiling to see if there's a bias for expression for one versus the other?
Yeah, absolutely. That is actually underway right now, the transcriptomics. And so we're really excited to see what parent is being more expressed than the other. I will also note that in the follow-up studies that we're doing right now, we're also doing some long-read genome sequencing. So these previous genomes were all short-read, both paired end and mate pair, but the times have changed, and we're really excited about having some long-read data to further tease out what's happening in these hybrids.
Fantastic. Look forward to seeing that. Jessica, do you have any more questions for Bertrand?
Yes, we do. So a question from Matt Blanco is, how difficult would it be to apply your approach to other pathogens or organisms?
I think that if any organism, especially with regards to the fungus, if it has a whole genome, then it's not that difficult, I think, yeah. As long as you have the whole genome sequence already, it's not a problem, I think. Because, yeah, it's been developed for other organisms, such as, I think probably those who work on Aspergillus know about the strap assay. So it's more or less the same technology.
Thank you. And then a question from me, and that is, can you give us an idea of what the medical laboratory resources would look like in places where fungal mycetoma would commonly be diagnosed? So how can your assay adapt to those sorts of conditions? And what advice might you have for people who are interested in assay development to keep in mind in applying these types of things?
I think this assay is not actually developed for routine use. So it's more or less, yeah, for a reference center. Because if you, yeah, where mycetoma is mostly sort of prevalent or endemic, it's like rural areas, more or less, with no health facilities. If I take, for example, Sudan, it's most of the patients come from like faraway provinces, from tiny rural areas with no access to medical care. So most of the time they have to travel to the capital, Khartoum, before they can see some sort of specialist center. So I don't think this assay is for routine use, but more or less for research and epidemiological purposes. Yeah, because in endemic areas, most of the time, what's happening, diagnosis is mostly often by yeah, traditional methods like culture and yeah, histopathology. At the moment, also part of our work involved we trying to develop a point of care test, which would help in diagnosis, but at the moment it's still preliminary.
Great. Any other questions? Feel free to either enter them into the chat or turn on your camera and microphone and ask them.
Bertrand, I had a question. Do you have any information about the clinical presentations of these different the different isolates that you had? I know you said there wasn't disease severity, but I think one of the strangest things about these mycetomas, you showed a picture of a patient who has this, and that there's no pain associated with this, that this is a completely that the fungus produces something that limits the amount of pain that the patient feels. Do you have any information about whether or not there's different experiences for patients who have these diseases?
I think most often the reason why patients come to the clinic with an appearance like what I showed is because of the painless nature of the lesions, because most of them don't report pain. From the stories I read and from my discussions with people in Sudan, they always say what the patients often report is like, oh, I was in the field or in the farm, and then I got pricked by a thorn, and then I came home. It was like probably started like a little fistule, and yeah, so I didn't think it was a problem. It wasn't painful, but then suddenly it starts to grow bigger. But then they also, the sad thing is then they start to use like herbal medication, and yeah, so by the time they come to the clinic, yeah, it's the feet is already in that situation where you have mixed bacterial infection, and also, yeah, most of the time I think like yeah, 56% of the patients end up with some sort of amputation. So it's yeah, but all of them report the beginning with no pain. Yeah, like yeah, it starts to grow. The tumor starts to grow with you don't feel pain.
A need for public awareness as well as clinical treatments, right?
Yeah, yeah. Probably just to mention that the treatment duration is quite long. I think it take yeah, it's treated with itraconazole 400 milligrams for like you take it for a minimum of 6 months, and yeah. Most often, first when you come to the clinic, if you diagnose, you take it for like 6 months, and then you come back because at first the itraconazole also sort of helps the lesion to encapsulate. for surgical removal or if it needs an amputation. And then following surgical removal, then you continue again with some sort of 6 months of treatment before you can have closure. So it's yeah, really a very debilitating disease, because it's the flesh. If it's not treated on time, bone involvement, and the only solution is an amputation.
Wow. Okay, thank you so much.
Yeah, many thanks to both our speakers. I think both talks were fantastic. So if you have questions, feel free to stick around. Otherwise, I hope to see everyone for our next seminar, which is May 13th.
Yeah, thank you, everyone. Thanks again.
Yeah, thank you, everyone.
Organizers.
Have a nice evening, morning, and afternoon, wherever you can be.